Hi, myself Devasis. I am the research head of Bionaval Life Science Private Limited, Bangalore. I work in different sectors of biotechnology such as molecular biology, microbiology, environmental biotechnology, enzyme technology, computer-aided drug designing, bioinformatics, etc. I am into different fields of product development as well as into optimization of different resources as well as calibration of instruments. So, before going into the research subject, I want to just give a thorough knowledge of what we have studied in the before session. The name of the session was mathematical expression of exponential growth phase. As of you will know that exponential growth is a fundamental in various scientific fields, particularly in biology, ecology and demography. It describes a process where a population growth rate is proportional to current size, leading to rapid increase in population over time. The mathematical expression of exponential growth provides a precise way to model and understand this phenomena, offering insight into population dynamics, resource consumption and potential impact on ecosystem and environment. So, before describing the entity of growth, we need to learn what is the equation, what is the representation of the population size, what is the representation of initial population size. So, there is a formula. So, population size at time t is calibrated based upon the initial population size at time t equal to 0. So, this population size depends upon the rate of increase or the growth rate of the microorganisms. So, in general the equation can be deduced as number of population at time t is equal to number of initial population size at the initial state or time 0 E into to the power r into t, where r represent the intrinsic rate of growth or intrinsic rate of increase of growth rate, and t is the time that is used for the population size that is estimated at the time t. So, the equation assumes a growth rate, a constant growth rate, where there is reasonable approximation in the environment, where resources are unlimited and conditions are optimal. So, the term e to the power r t signifies how the population multiplies over time, emphasizing the exponential nature of the growth. Now, let us discuss what are the biological implications of the mathematical expression of exponential growth phase. As we all know, in biology, exponential growth is often observed in microbial population such as bacteria, fungus, etc. So, this growth during the early phase of colonization in the new environment is required. So, here what happens when there is a bacteria in the new environment, there is abundance of nutrients and there is still a minimal amount of competition. So, what happens is that the organisms can reproduce rapidly. For example, if you see a single bacteria at this exponential growth can divide at every 20 minutes, this division can give rise to over a million of descendants in a very few hours. So, this phase is critical in understanding the spread of the infectious disease. The initial exponential increase in the pathogen number can lead to outbreak pandemicity, underscoring the importance of early intervention. Public health strategy often aim to reduce this rate, this intrinsic rate of increase through vaccination, quarantinization or treatment to control the spread of the disease, which you see in India during the COVID-19 period. What we have done? We have applied some public health strategies. We have quarantinized the individuals, those who are coming from other countries, which we thought of being infected by COVID-19 virus. So, this one helped in the control of the spread of the COVID-19. Then coming to the ecological and environmental context. In ecological context, exponential growth is typically observed in invasive species. So, what is an invasive species? So, these species are foreign to that environment. They invade the local flora, the local fauna, the local biotic resource, the local abiotic resource. So, these are generally newly introduced populations into the environment. So, these species can exploit the natural resource, as because they do not have or they lack the natural predator or competitors. This leads to unchecked growth. However, this phase is often temporary, as environmental resistance factors such as limited resource and increased competition eventually curb the growth rate, leading to logistic growth models. The exponential growth model also serves as the benchmark for understanding the deviation in real world scenario. For instance, the human population has exhibited exponential growth over the past century, driven by advances in medicine, agriculture and technology. However, concerns about sustainability and resource depletion highlights the limitation of perpetual exponential growth. So, the mathematical expression of the exponential growth phase is a powerful technique or tool in scientific research. It provides a foundational framework for understanding population dynamics under optimal conditions. While real world scenario often require more complex models, the exponential growth equation remains a critical starting point for studying growth processes across various disciplines. So, basically the implication extends from microbial ecology to human demographics, highlighting both the exponential and the challenges of exponential growth in nature and controlled environments. So, let us discuss the current chapter, measurement of growth and growth yield. The subjects that we are going to discuss in this chapter are the concept of microbial growth, the quantification of the microbial world, how we are counting the microbial world, then the growth yield, measurement of microbial growth, where we are adopting different techniques, OD measurement method, cell viability method and biomass determination method, and that significance of these methods for the study of microbial growth. Thirdly, we will be learning about the measurement of different growth yields, and at last the application of this growth measurement in the real world scenario. So, the first point we should discuss, what is measurement of growth and growth yield. Microbial growth, as the name suggests, represent the increase in number of microbial cells in a population with increase in time. As the time increases, the population also increases. The growth process in microbiology plays a vital role in various fields of application, that includes biotechnology, medicines, agriculture and environmental science. So, the quantification or counting the number of microbial cells or limiting to the factor that microbial count is the essential parameter to understand the dynamics of microbial population, understanding the critical attributes for an industrial process and optimization of processes, thereby helping in evaluating the efficiency of production, purification and downstream process. Growth yield can termed as the amount of biomass or product that are produced per unit of substrate consumed. So, this is one of the critical parameter in the microbial studies. So, the measurement of microbial growth is the choice depends on the specific objective of the study that characterize of the microorganism being analyzed and the available resources. So, microbial growth can be determined and quantified by various methodologies. Number one methodology, that is the most important part, is optical density measurement. The second one is viable cell count and the third one is determination of the biomass of the microbial cell colonies. So, the optical density measurement is suitable for high throughput screening of microbial growth in laboratory settings, while the viable cell count is preferred for accessing cell viability and enumeration of colony forming units, or it is called as CFU per ml. The biomass determination method are valuable for accurately quantifying cellular biomass in the bioprocess and industrial application. Most prominent method for measurement of microbial growth is optical density method. This method utilizes the absorption of light by a bacterial or fungal cell mass that are determined and quantified by spectrophotometric method by the measurement of the cell turbidity of a culture at a certain wavelength. Especially for the bacterial cell, we go for 600 nanometer. For fungal cells, we go for from 400 to 420 nanometers, that is called the lambda max. The principle behind the optical density method is that the turbidity of the culture increases with growth and multiplication of microbial cells, while the OD measurement provide a rapid and non-destructive means of accessing microbial growth. It has certain limitations that includes the inability to distinguish between live and dead cells and its susceptibility to interference from non-cellular particles or extracellular substances. The second method to determine microbial growth is viable cell count method. Viable cell count involves spread plating of a dilution of a culture onto a nutrient agar media. So, you have to first spread a particular dilution, maybe 10 to the minus 4, 10 to the power minus 5, 10 to the power minus 6 according to your selections onto a nutrient agar media, and then you incubate that nutrient agar media at a desired temperature and a time in a bacteriological incubator. Basically, the desired temperature is at 37 degrees centigrade and almost within 24 to 36 hours we get the number of colonies. Then afterwards what we do? We go for physical examination of the cell growth. We determine different cell morphology. That cell morphology can include the cell shape, the margins, the color, the morphologies, whatever you can see visually, and then count the colony forming units to determine the number of viable cells. So, these viable cells can also be determined and visualized by adapting methods such as microscopic measurement, acridine orange direct colony counting, 4,6 diamidino to phenylindole direct counting, also called as DAPI counting. And there is another viability kit method, that is called the live dead backlight bacterial viability kit. Another very simple methodology that we use during the cell viability method is the use of even blue. So, even blue is commonly used to assess the viability of the bacterial population in conjugation with other strains, such as fluorescent dye like PI called as propidium iodide. In this application, live cells exclude Evans blue and PI, okay, while the dead cells take off the even blue and PI. So, this is the differentiation of live and dead bacteria or live or dead population. Okay. So, the viable cell count method is particularly valuable for accessing the viability and culture availability of the microbial population, especially clinical microbiology and food safety application. However, sometimes these methods require longer incubation time due to underestimated cell counts, when the culture containing the cells do not form colony under the chosen conditions. The third method to determine microbial mass or microbial growth is biomass determination. So, let us first see or know what is a biomass. So, biomass, as the name suggests, is the overall mass of a substance. Like you are an organism, so you have certain mass. So, that mass contribute to your biomass. So, biomass determination method are of basically three types what we determine in the microbial growth. The first one is the dry weight measurement. The second one is the content of the protein, that is protein content analysis. Third one is the amount of nucleic acid content in the organism, that is called nucleic acid content analysis. So, biomass determination is one of the direct method that uses to quantify the amount of cellular material present in a culture. So, dry weight measurement involves filtration of the culture, followed by drying of the filter paper and then weighing the residual biomass to get the dry weight. So, it is just the amount of cells that we get after removing from the culture media. The second one, protein content analysis. Protein content analysis is another method that is employed, which utilizes spectrophotometric or colorimetric assay, such as Bradford or Lorry method, to quantify or estimate the concentration of the protein in the biomass. Quantification of nucleic acid is another method that employs quantification by spectrophotometric method to access the concentration of the nucleic acid, may be DNA or RNA, in the biomass. So, biomass determination method provide accurate measurement for cellular biomass and microbial growth, but these methods are labor intensive, require specialized equipment, high precision analytical instruments and high grade chemical reagents. So, what is the significance of growth measurement? The significance of growth measurement has application in diverse scientific disciplines and industrial applications. The accurate determination of microbial growth gives supportive insight to microbial physiology, ecosystem dynamics, technological process and environmental interactions. In microbiology, growth measurement helps in characterization of microbial strains, assessment of antimicrobial activity, determination of growth attributes, etc. So, by employing sophisticated measurement techniques and methodology, a researcher can unravel the complexity of microbial growth and harness its potential for addressing global challenges in health, environment and sustainability. In ecology, the growth data are used to study and monitor population dynamics, trophic interactions and ecosystem productivity. So, growth measurements also play crucial role in optimization of bioprocess for the production of biofuels, pharmaceuticals and industrial enzymes. So, in general, the continued innovation in growth measurement approaches will further enhance our understanding of microbial systems and facilitate the development of innovative solutions to complex scientific and technological problems. Now, how to measure the growth yields? So, growth yield referred to the ability and efficiency at which a microorganism can convert substrate into desired products and biomass. So, commonly used measurement of growth yield or quantification of the growth yield includes biomass yield, substrate yield and product yield. These are the three main factors or measurements for the growth yields. It can also be defined as the amount of biomass produced per unit of substrate consumed. So, measurement of growth yield provide valuable insight into the efficiency of microbial metabolism and nutrient utilization. So, understanding growth yield can aid in optimization of processes for various applications that includes biofuel production, bioremediation and pharmaceutical manufacturing. The first measurement for growth yield is biomass yield. So, this biomass yield is typically expressed as the amount of cellular biomass produced per unit of substrate consumed. So, this parameter provides the insight into the efficiency of cellular metabolism, energy conservation mechanism and nutrient utilization pathways. Biomass yield measurement is one of the fundamental aspect for the evaluation of performance for different microbial stress under various growth conditions. This also includes optimization of bioprocess for biomass production. By quantification of biomass yield, researchers can determine the effectiveness of different culture strategies, optimization for media composition and most important part is the genetic modifications to enhance microbial growth and productivity. The second measurement for growth yield is substrate yield. So, substrate yield is another critical aspect for growth yield measurement that quantifies the efficiency for conversion of the substrate into the product mass. It represents the ability of the microbes to utilize available nutrients for growth and metabolic activities. Substrate yield calculations involve the ratio of biomass produced to the amount of substrate consumed, typically expressed in terms of gram of biomass per gram of substrate utilized. Again I am repeating, it is expressed in term of gram of biomass per gram of substrate. So, understanding the substrate yield is essential for optimizing the bioprocess mechanism in industries. Such industries are intended for production of biofuels, those are intended for bioremediation process, and especially the fermentation based manufacturing industries. In addition to biomass and substrate yield, product yield can also be a crucial parameter in growth yield measurement, particularly in biotechnological application when the production of specific compound or metabolites is desired. Product yield quantifies the efficiency of formation of products from the substrate and reflects overall performance of microbial strains in synthesizing target compounds of interest. Whether it be the production of biofuels, pharmaceutical products, enzymes or special chemicals, optimizing product yield is the paramount for commercial viability and process scalability. Then coming to another factor, the application of growth measurement. The accurate measurement of microbial growth and growth yield can have diverse applications across various scientific disciplines and industries. In microbiology, growth measurements are utilized in clinical diagnostics, food microbiology and microbial ecology. In ecology, growth data are integral for studying population dynamics, community structures and nutrient cycles. In biotechnology, growth measurement play a critical role in process optimization as well as improvement of the strains and product development. Moreover, the growth measurement are essential in environmental science for accessing microbial activity, pollutant degradation as well as ecosystem health. Then coming to what are the challenges and future directions. While the significant process or progresses has been made in the measurement of microbial growth and growth yield, several challenges remain. Standardization of measurement techniques, particularly in complex environment, is essential for ensuring reproducibility and comparability of the results. Furthermore, advancement in high throughput technologies and computational modeling hold promise for enhancing the accuracy and efficiency of growth measurement. Future research direction may focus on integrating multi-omics approaches to gain comprehensive understanding of microbial growth dynamics and elucidate complex metabolic network. So, let us summarize the whole thing. The measurement of microbial growth and growth yield is fundamental in various scientific disciplines and industrial applications. Accurate quantification of growth dynamics provide insight into microbial physiology, ecosystem process and biotechnological process. So, by employing a diverse array of measurement technique and methodologies, researcher can unravel the complexity of microbial growth and harness its potential for addressing global challenges in health, environment and sustainability. So, here we will discuss what are the topics that will be in the next lecture. So, your next lecture that follows this topic are techniques of pure culture. So, let me introduce some techniques or some agenda what you will learn in the next topic. So, pure culture technique are fundamental in microbiology for isolating and studying single species of microorganism without contamination from other species. This technique enable a researcher to investigate the physiological, genetic and biochemical characteristics of specific microbes in a controlled environment. So, one method is that the stick plate method, that is widely used for its simplicity and effectiveness in obtaining isolated colonies from a mixed sample by progressively diluting the inoculum across a agar surface. The pour plate method is another methodology that involves diluting the sample and mixing with molten agar and to isolate those colonies within the medium, while the spread plate method is another technique where you spread a diluted sample evenly across the surface of the agar for the growth of the colonies. So, selective media can also be used as because they inhibit the growth of unwanted microbes, facilitating the isolation of specific microorganisms. Some advanced technology or techniques like micro manipulations and flow cytometry allow for precise single cell isolation. So, by mastering these techniques are very crucial for the advancement in microbiological research, clinical diagnostics and biotechnology.